Hi everyone, welcome to Raaonline. In today's lecture, we will be talking about the infections caused by Shigella. What is Shigellosis? Okay, I am Dr. Priyadarshini Shanmugam, Professor of Microbiology. And we will be talking about Shigella under the following headings. Okay, what is the definition of Shigellosis? A classification of the genus Shigella. What is the pathogenesis of Shigellosis? The clinical features, the lab diagnosis, and then a few differentiating points between bacillary dysentery and amoebic dysentery. Okay, so when we go on to Shigellosis, it means it's a diarrheal infection which is caused by Shigella species. So it's an infection of the gut. It causes a diarrhea, but more importantly, the Shigella causes dysentery. So dysentery is different in the way it means that it also, in addition to there being diarrhea, the person also passes blood and mucus in the stool. So whenever you say dysentery, it means a diarrhea accompanied by blood and mucus. Okay, and the person also has abdominal pain as well as tenesmus. Okay, and this is mainly caused by bacteria which belong to the genus Shigella. And Shigella are identified as gram-negative bacilli, that is rod-shaped short bacteria. They are non-spore forming, they are facultative anaerobes, and they infect the intestinal lining. And Shigella are spread by the fecal-oral route of transmission. And Shigellosis is an important public health problem not only in developing countries but also in the developed countries. Okay, and the most important agent, that is Shigella, and this genus name was got from the name of a Japanese microbiologist, Kiyoshi Shiga. Okay, so his name was Shiga. So the organism, the genus is named after him as Shigella. And this Kiyoshi Shiga was the microbiologist who isolated the first member, Shigella dysenteriae, okay, serotype 1. It's also known as Shiga bacillus or Shigella shige. And this was isolated as early as 1896 from epidemic dysentery patients. And the problem with the Shigella is that there is an increasing resistance being noted, okay, by this organism. This organism is exhibiting increasing drug resistance to first line and second line antimicrobial agents. So that is a problem which we need to be worried about. Regarding the classification of Shigella, they belong to the family Enterobacteriales. Earlier this was known as Enterobacteriaceae, now it's known as Enterobacteriales. And in this genus is Shigella. And based on differences in biochemical properties as well as serological, that means the antigenic properties, mainly relating to the O antigen, the Shigella are classified into 4 species. Okay, Shigella dysenteriae, Shigella flexneri, Shigella boydii, and Shigella sonnei. Out of which Shigella flexneri is the most common species in our country, that is India, followed by Shigella dysenteriae. Based on this O antigen, I told you they are classified into different species, and again based on the O antigen, each species, each of these 4 species has got several subtypes. So Shigella dysenteriae, if you see, it has got 15 serotypes based on the O antigen subtype. Flexneri has got 8 and boydii has got 19 serotypes. But if you see Shigella sonnei, it's antigenically homogeneous, meaning antigenically only one O antigenic type of Shigella sonnei is present. So only one serotype is present, but it can be typed further by using something known as collicin typing, okay, into 26 collicin types. And regarding the pathogenesis of Shigella, we need to know that this is the most important pathogen causing bacillary dysentery. And as I told you, dysentery means increased frequency, okay, and then there is diarrhea, but it is accompanied by blood as well as mucus in the stool. And the mode of transmission of Shigella, as for any GIT infection, it is through ingestion. Either you can contaminate your fingers and then transmit the infection to yourself, or it can be through contaminated food, water, very rarely flies. It can also be transmitted sexually. Homosexuals are at increased risk of developing Shigellosis. And this bacteria is highly infective. So the minimum infective dose, in fact, as long as less as 10 bacilli to 100 bacilli is enough. If you ingest 10 to 100 bacilli itself, it is enough to cause an infection, because these bacilli can survive the stomach acidity. So the acidity present in the stomach is not enough to kill them. So even if 10 bacilli are ingested, they can multiply and they can cause the disease. So that is one thing we have to enter. So continuing the pathogenesis of shigellosis, I told you it enters into our alimentary canal via the fecal-oral route. So by ingestion of contaminated food. So once it has entered into the stomach, it passes through the gastric acid and then it enters into the intestine, where the mucosa is targeted, and in the mucosa you have some special cells known as M cells, okay, and these bacteria enter into the M cells and then they are engulfed by the macrophages, which are staying in close association with these M cells. Then these macrophages release the bacteria, and once these bacteria get released by the macrophages, you will be having other inflammatory cells like our neutrophils, that is the pus cells. They will move to the infected sites, and then because these pus cells are also moving there, a lot of cytokines are released, and these cytokines only cause the gut to get inflamed. So that is only acute colitis, which is the hallmark of shigellosis. Okay, so all this invasion into the cell and everything is made possible because the Shigella has got a very large plasmid. It's a virulence determining plasmid. So it's a virulence plasmid, and the same kind of plasmid is seen in enteroinvasive E. coli. So not only Shigella but also EIEC has got this large virulence plasmid. So now let's see this picture. So the Shigella are entering into the intestine, and these are the M cells. They attach, and then they enter into the M cells. From there, they are taken up by the macrophages. So what is this M cell? M stands for membranous or microfold cells. So they are an important subset of the intestinal epithelial cell, or IEC, okay, intestinal epithelial cells, and they are responsible for immune sensing of all the luminal bacteria, okay. So they sense that these bacteria are present and see how it gets internalized, and then it goes into the macrophages. Then the macrophages release them. As the macrophages release them, you also see that these interleukins are also released. So these interleukins will recruit the other inflammatory cells like your polymorphonuclear cells and all the other pus cells, okay. So more cytokines. So as a result of which all this gets inflamed. But in addition to it, I told you Shigella has got this virulence plasmid, and this virulence plasmid only produces certain proteins. I will tell you what they are. They will enable it to enter inside the epithelial cells, and also it will cause damage to the epithelial cell and also spread from cell to cell. So it enters, it damages, it multiplies, and then it spreads into another cell.